incredibly bad.
From Microbe TV, this is TWiEVO, This Week in Evolution, episode 108, recorded on November 20, 2024. I'm Vincent Racaniello, and joining me today from Salt Lake City, Nels LD.
Hey there, Vincent. Great to be with you from LD Lab Studios. And
looks bright there.
Yeah, it's bright. The sun is out. There's a little snow in the mountains, and
Nice.
My computer is working. So it came in a little bit last minute here. Did a quick restart.
Don't you have like 10 backups, Nels?
Yeah, I should. I have about 100 applications open, and
that's the problem, right?
Tabs at any time, and so we're kind of stumbling out. I am stumbling out of the blocks here, but great to be here. Great to be together, and lots to talk about. Did anything happen, Vincent, in the last 3 weeks since we were last podcasting together?
It's like the world has changed.
Oh my goodness. Yeah. Feels like it.
You know, here on TWiEVO and Microbe TV, we are pro-science. We recognize that science makes everyone's lives better. So try taking it away. I'm sure everybody knows what we are talking about. Speaking of everybody, welcome, folks, to another episode of TWiEVO, This Week in Evolution, which is now live streamed every episode. It's the only podcast that we do that way. It's cool.
Yeah.
And welcome our moderators. We have here Barb Mack.
Welcome.
And Les. Thank you for joining us. Tell us where you're from, folks. We'll say hello before we get started with the science. Amy is here. Welcome, Amy.
Oh, Amy, good to see you.
Good to see you. And Amy reminds us about the Microbe TV fundraiser channel. Your inner scientist. Join the Potter family and your moderator, Tom Steinberg, with your video audio of how Microbe TV has empowered you. So we're asking you to send in a little video or audio you can make on your phone. Just tell us what we mean to you, right? Empower your inner scientist, and we'll use it in our fundraising efforts. We've gotten ones from Tom, our moderator, and
awesome.
The Potter family.
That's great.
Check that out.
Check that out. And I just want to say, Vincent, not only kind of for fundraising purposes, but also Microbe TV morale, hearing from you and how this might be a positive part of your daily routine or weekly routine or monthly routine can really put a lot of wind in our sails. I still think about this letter we got years ago, Vincent, from Tia in Virginia, and how TWiEVO is useful in her science-related homeschooling efforts, and that still helps to sort of sustain my energy when thinking about what we're doing here. Also, your point on the live stream, I think some great advantages of it is that we are able to interact in real time, thanks in large part, again, to our moderators who sort of keep things organized along the ways. One of the things that's done that I've been thinking about and wanted to talk with you, Vincent, is, it's a lot of you and me discussing stuff, because sometimes it can be tricky to get a guest onto a live stream, sort of a whole talking without a safety net sort of scenario. But I'm thinking about putting out sort of a all points bulletin soon to just see if there are colleagues, science curious folks who want to propose joining us for a conversation. So stay tuned. I'm going to put that out there and, see if we can grab someone kind of at our regularly scheduled times to see if there are folks who want to sort of kick around some science with us as it relates to evolution.
We need to have more science communication, because it's going to be a tough 4 years for science. As you know, RFK Jr. has already said he wants to cut infectious disease research for 8 years. Where are you living under a rock? Oh my God. So we have to keep you thinking about science with Microbe TV give you ways to fight back. And the more of our colleagues who come and talk about their work, the better.
Exactly.
Because they can come and wow people, and they say, this is infectious disease research.
That's
and
right.
This is what's been going on for years, and it's great stuff, like even today's paper.
Exactly. So that was what motivated the choice of our preprint that we'll get to in a few minutes here, was to try to lift up some infectious disease research and some surprises about how sort of searching in corners that you might not expect, interesting things can sort of show up and sort of start having these butterfly effects, these ripple effects that really drive discovery, drive innovation. And yeah, this is a moment for very obvious reasons that telling the stories of science and, sort of in our corners of the world, the stories of infectious disease, of viruses, parasites, other microbial critters is obviously as crucial as ever. A little bit in that space, Vincent, I wanted to say that I'm hosting the author, someone we've talked to on TWiEVO before. David Quammen will be here in Salt Lake on December 10th, giving a seminar on the ecology and evolution of what he's describing as scary viruses. So this is the author of Spillover, I think up to 17 books now. We spoke about one of his books about evolutionary trees,
the tangled tree, right?
The tangled tree, exactly. And so he'll be here. Now the theme that we'll be discussing is, that viruses, for example, could really use storytellers. These powerful genetic elements that move through populations in obvious ways, but invisibly, and how that really can kind of the human impulse to sort of direct what's invisible to human actions versus what's really happening in biological reality, and to try to sort of lift that up and also strategize. How do we tell better stories about the infectious microbes around us and the people like us who study them? And so lots of work to do, and feeling like this is a great time to invest time and energy in that space.
That should be fun. He's great.
Yeah.
That'd be a lot of fun. Okay, let's see who's here.
Yeah.
Philip says, interesting times to be alive in. Oh, yes. I'd rather it not be interesting for these reasons, but for reasons of cool science, for example, but it's what we have.
Yeah.
Let's see who else is here.
Philip, glad you're here.
Barb Mack is here from the UK, of course, one of our moderators. Thanks for coming. Glass is here from California, another one of our moderators. Thank you for doing that.
Yes.
Abdul Aziz is from KSA, Kingdom of Saudi Arabia. Welcome.
Welcome back.
Zombie whatever 2.0 is from Eastern Pennsylvania. And by the way, Philip is from Wales, UK.
Fantastic.
Who else? Muiza is a student in Zambia. Cool. Doing clinical medicine.
Cool.
Welcome.
Welcome.
Good
Glad
to
you're
have
here.
That's it. Well, there are other people, but they didn't tell us where they're from. But that's okay.
That's all right.
By the way, we have a link for you. I'm going to... Let's see. So, a nonprofit organization called SafeCommunitiesCoalition.org contacted me the other day. They're a 501c3, and they made a website where you can go and write a letter and hit the button, and it goes to all the senators. Mm-hmm. So if you would like to oppose the nomination of RFK Jr. as the head of Health and Human Services, you have to be in the U.S. to do that. So you would have a senator in your state, 2 senators.
Yeah. You
can go to I'm going to type this in the chat. It's www.safecommunitiescoalition.org/RFK. They have a special page just for this dude. I think he knows something about science.
Yeah.
And we've also put out a video, which I put the link in earlier, a 5-minute video with Paul Offit debunking like 5 different things that he has said
Yeah,
For
fantastic.
example, Nels, he
Yes.
said no vaccine has ever been placebo controlled.
Mm,
He said the polio vaccine has killed more people than it has helped.
mm, yeah.
He said the 1918 flu pandemic was caused by the flu vaccine.
Mm.
So, Nels, I don't understand how someone can get away with this falsification.
Yeah.
I don't care if it's knowing or unknowing, and then be nominated for a very important position. I just don't get it. The way I grew up was
Yes.
you earned your position by knowing your field, you know?
Correct. Yeah. No, a lot to talk about. So first of all, that link, thank you for sending that, Vincent. I um, followed through on that myself. I mean, these are like not even drops in the bucket of sort of responses and sort of the hurricane of whatever is happening at the moment. I mean, so I've been thinking about this kind of in terms of like almost an unnatural disaster. Like it feels like a hurricane is moving through right now, sort of in real time, and it's all the chaos, all the instability, all the noise, and trying to I think there's this almost these on this two levels, as if we're supposed to like, so the statements, the sort of misinformation or disinformation that you, those examples you gave, like we're supposed to squint and say, oh well, he didn't really mean it somehow, or things like that. But I agree with you. I mean, the idea, just in addition to sort of the impulses of misinformation or throwing all this sand in the air, just like character, like someone who would take a roadkill bear and put it in Central Park to make it look like the bear was hit by a bike accident, as if that's cute or interesting or funny. I mean, um, this is disastrous in a moment where, politically, some of these nominees are sort of like, we'll get a few through if you compare it to some that are having sex parties with 17-year-olds as sort of a regular sort of social scenario. And so, that's just so over the top and out of balance in so many ways that it's just a chaos. That doesn't strike me as a sort of a conservative approach, sort of in the historical framing of this political party, that, chaos, instability. I don't know what's signal and what's noise, what's supposed to be some cute joke, or what's really going to sort of propagate as policy. And the ability to, just break stuff, break shit, is relatively easy in comparison to building something. And so when a hurricane, like in this case, an unnatural hurricane moves through, to extend the analogy, if you knock over a bunch of trees, that's going to take decades to replant. And I get it. There's other sort of viewpoints out there that, no, we want to get rid of those trees and build a highway through that, what was sort of more natural habitat, or however it might go. But wow, in this moment of thinking about what's at risk and what's vulnerable, and how to stand up for those things, or to communicate the importance of them, in the hopes that you mitigate a disaster, you put up the storm windows, or the but let's be straightforward about what's going on here. It's a disaster unfolding before
I
us.
so I mean, HHS, Health and Human Services, you need someone who's qualified. RFK Jr. is
Yeah.
clearly not. You want someone morally correct in that position, right?
Mm.
I don't understand what the point of all these ridiculous, and there are other nominations, a TV co-host,
Mm-hmm.
right?
Yeah.
They're all ridiculous nominations. What is the intent, to break the system? Is that it? I don't know.
Feels like that's part of it. I mean, it feels like there's similar feelings popping up from January 6th, 2020, one, when, like, just sort of that action, that chaos, in a very vivid area, which is now, trying to be, or not trying, is being recast as, a celebration. I saw someone made the comment, like, is it possible that January 6th will be proposed as a national holiday? And sort of completely, turning things upside down about how we think about what the United States is. I mean, this is an unnatural disaster unfolding in ways that are still really unclear and pretty frightening in a lot of ways, to say the
I
least.
just got this Instagram post. It's a picture of a young Tony Fauci there, and
Yeah.
it says, "Sure, I spent 50 years of my life studying viruses just so I could trick rednecks into wearing paper masks. That was my career goal." You know, if you think about things, folks, and you throw away your emotions and hatred and bigotry and just step back and think of things like, yeah, we're trying to trick you into wearing masks. We don't need infectious disease research. We don't need vaccines. Think about it. Use your brain and you can realize it's not right. Oh my
Well,
God.
it is interesting. I mean, so, it's not new that people's experiences influence governmental outcomes. So I'm thinking about, I don't remember if this is exactly 15 years ago, but, Arlen Specter, who was a
Yep.
senator in Pennsylvania, I think, was
Yep.
diagnosed with cancer. And that, given his political connections and sort of the way things have worked, that actually meant a pretty sizable infusion of resources into cancer research in a positive way. And so, um, you know, um, Tony Fauci recently, I think, had a West Nile virus infection, speaking of sort of personal encounters with infectious disease, which was really harrowing. And, there's going to be a lot more, depending on how the direction things go. Still very sort of all of this kind of forecasting and all this confusion and chaos. But there'll be a lot more infectious disease. And as humans grapple with that, especially ones who are sort of in political positions of power, there might be some unfortunate lessons coming down the line that could be avoidable.
Just think of an example. If he had been installed 4 years ago as HHS, right?
Yeah.
He would have discontinued infectious disease research. We never would have been able to make a COVID vaccine because we didn't have any infectious disease research. That was all done at NIH, which he wants to decimate. He wants to fire 600 people from NIH and cut infectious disease research entirely. So we would have no COVID vaccines. And don't any single person tell me COVID doesn't matter. If you think that, you're just lying, okay? Because it's not true. Millions of people have died. Hundreds of millions of people have been infected. We needed those vaccines. And so if you discontinue infectious disease research, as Nels just said, pathogens are everywhere. We don't know where the next one is coming from. So what if he's in charge and we have an H5N1 pandemic? We're not going to be doing any research, so we can't deal with it. You want people to die. So we could wait for a disaster to happen, or we could not confirm him. I think that would be good. The only thing now is if you don't confirm him, some other wacko is going to be put into his place.
Well, yeah, that's the thing here is part of the strategy is all of this noise and chaos. And then some sneak through, some don't, but then who's behind that? And then there's a whole nother sort of pathway of stuff, right, going along behind the scenes. It does seem clear to me that part of the strategy here is to, by saying these provocative things, almost this, sort of, it just, for good reasons, really hits a nerve with folks that have dedicated their careers to work in this area. And so how much of it's noise, how much of it's signal, it feels like we're in a middle of a natural disaster. The storm is arriving in waves.
I just want to say one thing. So if the goal is to break this country, is to break democracy and turn it into an autocracy, right?
Mm-hmm.
Many billionaires keep their money here in the U.S. because it's safe. You think they're going to keep doing that if it's a friggin' autocracy? No. So think twice about that, folks.
Yeah, for all the billionaires who are listeners of TWiEVO.
Yeah, right. There aren't any. I mean, but for our listeners here, that is a real thing, that people with a lot of money park it in democracies in general because they're stable. And in an oligarchy, the leader can't just take all your friggin' money. In Russia, they can just take all your money out of the banks.
Yeah.
Can't do that here.
We're in our community. I think we're here to grapple with all of this just noise and fury at the moment, and hanging in and hoping for the best.
Yep.
So I could keep going for hours on this topic, but we've got some science too, Vincent, that I think would be really fun to to lift up and discuss and get
For sure.
some dialogue going, some conversation going. So, speaking of infectious disease, today's paper, it's a preprint that just came out in September. The title is Virus Associated with the Zoonotic Pathogen Plasmodium knowlesi causing human malaria is a member of a diverse and unclassified viral taxon. So we've got two infectious microbes. One is Plasmodium knowlesi, rhymes with bullseye. I asked my colleague who's right next door, Ellen Leffler, who works on Plasmodium, the population genetics of it and human genetic variation that can lead to susceptibility or resistance to it. And so Plasmodium knowlesi. And this interesting idea put forward here. This is so Eddie Holmes and some collaborators and trainees in his group. So the authors are Mary Petrone, Justine Chiron, Matthew Grigg, Timothy William, Giri Rajaram, Jacob Westaway, Kim Piera, Meng Shi, Nicholas Anstey, and Eddie Holmes. And so the idea that they're in, by the way, international group. So the Holmes lab is headquartered in Sydney, Australia. He was at Penn State something like a decade ago and moved to Australia. And then some collaborators, both from China and several institutions who are really doing some interesting work related to, and we'll dig into this a little bit, related to using machine learning and artificial intelligence to try to interrogate, to search through metagenomic data sets. We'll talk about this in some more detail and pull out or discover sequences of viruses that haven't been seen before. And then finally, some collaborators from Malaysia. So Plasmodium knowlesi. So the most famous and impactful Plasmodium is Plasmodium falciparum, which causes most cases of malaria in humans. And don't have the numbers exactly at my fingertips, but we're talking globally. We're talking about on any sort of annual basis between 500,000 or plus or minus 100,000 deaths of humans per year. And something like 250 million new infections or infections that continue every year. And so here, the interesting idea they're putting forward is, if we discover the viruses of Plasmodium, so the parasites that cause malaria, can this give us some information about, or even sort of a foothold to think about interventions? And so, malaria has been largely or maybe temporarily, unfortunately, solved in the U.S. So malaria, Plasmodium, is vectored by mosquitoes.
That's an infectious disease, right?
Indeed, it is. It's something that you wouldn't want to take a 8-year break from putting resources into studying. I think it's safe to say. And so, with a lot of sort of environmental control measures, how we've controlled mosquito populations, which are incidentally encroaching back in new ways as environments are changing, habitats are changing. We're seeing some of the species of mosquitoes that are vectors to spread viruses and other diseases, parasitic diseases like Plasmodium. And one of the, sort of new emerging issues, or not even new, but one of the ongoing issues is that we have a few drugs to Plasmodium, but resistance evolves really quickly. And so, drugs and vaccines have had limited success so far, and resistant strains regularly arise. So, one of the kind of interesting ideas sort of put forward right away is that, thinking about sort of an analogy to bacteria, the viruses of bacteria called phage, which regularly infect lice, kill bacteria, has been pursued for decades as an alternate antibiotic strategy. And we've talked about this, Vincent. So we've got episode, let's see, where are my notes here? I'm a little disorganized today. TWiEVO 44 with Paul Turner, who's at Yale University. The title of that TWiEVO is "The Enemy of My Enemy is My Phage." And so here, the notion is that, and there's been some success, by the way, so antibiotic-resistant bacterial infections with patients who are close to dying, where taking a phage that specifically infects those pathogenic bacteria have saved the lives of humans. And so there's a lot of work, hopefully ongoing work, in the resources, ongoing resources, to continue to develop it. It's not so simple, but some really interesting. So this is TWiEVO, some really interesting evolutionary ideas here. So, in the case of phage, there are ones that target the receptors on the surface, the transporters on the surface of bacterial cells that pump antibiotics out of them. And so it's almost like a two-step process where the longer those bacteria see the phage, the ones that survive are the ones that get rid of or don't have those transporters. And so those antibiotic resistant bugs are now antibiotic sensitive. And so that's sort of the second step, or the second punch, is to, after the phage moves through, the viruses move through, then the remaining bacteria are actually, the pathogenic bacteria are actually antibiotic sensitive. You flip the switch on that using this sort of reasoning, evolutionary reasoning. So Paul Turner and others are advancing that in some really, I think, promising, interesting ways, but complicated. It's not just, you don't just sort of say that and snap your fingers and it works. There's a lot of research involved in trying to get this to go. So anyway,
this idea
yes,
that knowing about viruses of parasites can be useful. There's another example in Trichomonas. There are viruses of Trichomonas. They're double-stranded RNA viruses, and they make the infection worse because the double-stranded RNA is recognized by the innate sensors, and you get more inflammation as a consequence. So perhaps targeting the virus can help reduce disease. So that's the idea here as well.
Yeah, exactly. It can work in both directions, right? The virus could be a tool that you could deploy if it sort of knocks out that population or changes the genetics of that. Or, as you're pointing out, and there's some evidence that will sort of, I think limited, but with some evidence that will weigh that some of these infected Plasmodium populations, that actually the sort of linking that to the epidemiology or the infection outcomes, that can be worse. And so, in that case, the two pathogenic microbes are sort of teaming up in a sense, one on the host, and then that makes the host, which we become the host of, sort of in worse shape. Okay, so to kind of rewind the tape, do parasites have viruses? And honestly, we know vanishingly little about this. We're swimming in genetic data, metatranscriptomic data, all kinds of data sets you just go to the internet and gain access to. Quick shout out for the folks, including governments, federal agencies that maintain these databases. So Open Science, as people are generating this data, the availability, the more that's democratized or available, the more progress that can happen quickly as we make these discoveries. And so this is another one of those cases, Vincent, where sort of the things that we take for granted or just assume are sort of stable and, will be supported. Not always the case. And so when it comes to even the availability of the growing genetic data, this is something that's also feels like it's at risk in some real ways. Even more so than usual. Okay, so the real trick here is that, so as you pointed out, there have been these discoveries of viruses, and in fact, one in a different species of Plasmodium. So they're focusing on Plasmodium knowlesi, but there's another species called Plasmodium vivax. And one virus has been discovered there. It's called the Matryoshka RNA virus 1. And it's in this pretty currently mysterious class of RNA viruses, the mycoviruses. Really limited data on this. They're sort of, we've just sort of glimpsed at some sequences. And then this one case, it's been tied to another species of Plasmodium. This one, by the way, doesn't cause the same kind of pathology in humans. Humans can be infected by it. It moves between primates and other animals, but doesn't cause the symptoms of malaria, of malarial disease. So key challenge here that the authors highlight is, these viruses, these classes of viruses that we've just glimpsed at are super different from the ones that we already are familiar with, the ones we know, the polio viruses, influenza virus, SARS-2, et cetera. And so this is really kind of the frontier, the real edge of thinking about, we're familiar with the viruses of our species, but not of the parasites that show up in the clinic. And so this is also kind of then this kind of interesting paradox, I would say, where a lot of science, it really does depend on these databases of information and sort of developing new tools to pull out interesting viruses or other, signatures of the kinds of genes that they encode. And so, the danger is, in some sense, the more you depend on this, the more you sort of you have blinders on. You're sort of finding things within the sort of collection of what's already been discovered or what's already known. And so the work here, and by other groups, is really sort of trying to still use this as a foothold, but then to reach even farther and identify, using increasingly powerful computational tools, sequences that sort of we haven't been proposed as being parts of new classes of viruses. But it really does, like, this sort of worldwide, just continuing to search and sample and to share information is really vital to these enterprises. So in particular, we've got some new techniques that are sort of coming online. This one is in sort of just a preview. I think the energy of this paper is sort of this balance between these sort of global approaches, and this is going through tens of thousands of data sets and using software tools that are increasingly depending on machine learning or artificial intelligence, sort of the buzzword of the time here, in order to train the software to distinguish between sequences that might be coming from a virus and ones that are just out there sort of in the whole sort of collection of sequences that comprise these so-called metagenomic data sets. And so we'll kind of circle back on some of the global approaches, but this one is a really directed one. So in fact, they set out with only 18 data sets, which is still, like, in the history of science, this is still a massive enterprise. So the data sets, these are genetic sequence, so blood samples taken from 18 patients, human blood samples. These are patients that showed up in the clinic in Malaysia, and I think with
Plasmodium, right?
Correct. Yeah. So they had symptoms of malaria, or in other cases. So 6 of them are Knowles-I, 5 of them are Falciparum, the species that causes most cases. 7 were Vivax, which is still, and there can be co-infection scenarios as well that can unfold. And then they have 6 non-infected controls. And so, the motivation here is so from these blood samples, then the researchers have collected RNA and just sequenced everything. So you'll have RNA. So this is, the RNA from our cells, as our cells are going about their daily biological business and making more proteins through RNA intermediates. Those are all sequenced in there. The other viruses, or the constituents, right, where the microbiome, where we have more cells that are microbial than we do cells that are us. And so all kinds of sequences there. And then the reason for the comparison is to hope that there might be something unique or different in the patients that have specific Plasmodium infections that are missing in either the control samples or even the Plasmodium of a different species. And so this gets at sort of the host range, right, of the sort of virus of the parasite. And so allows them to make those comparisons.
So we should also point out that they find the Matryoshka virus in all 7 Vivax isolates. It's known to be a virus of Vivax, right?
Exactly. And so that's where it's really kind of useful to have a little bit of those, all of that data, those footholds, because you're kind of in that search. It's hard to sort of, so the way we write these papers is sort of like, oh yeah, we discovered this virus. It just sort of fell into our lap out of these sequences. But in reality, you're sort of faced with this like mountain of data. There's no way you're looking at it. This is all through computer-based searching. And they use a tool called the Diamond Blast Search, which is already an established tool that is very good at, or it's increasingly useful in the sense that it can kind of go through all of these sequences and say to you, you might want to look at this one. And we'll get to sort of the details in this in a moment. But you're going to find a little bit of everything. And so having those, sort of guideposts along the way are really valuable. And so just like you're saying, Vincent, finding that other one and that it's specifically in the Vivax samples, not the other, that's sort of giving you some confidence by comparison that the things that you're seeing are real. But most of it's just like junk out there. It's really an interesting and ongoing challenge. Okay, so the thing that they're searching for here is something that's pretty specific, not completely, but pretty specific to RNA viruses. And these are sequences of the gene sequences encoding the protein RDRP, RNA-dependent RNA polymerase. So, Vincent, we got to talk about this polymerase because it's pretty central, a hallmark
Oh,
of RNA
I just
viruses.
I just revised the chapter in the textbook on RNA polymerases.
Oh, wow. Yeah,
RDRP is the only protein universally conserved among all RNA viruses.
exactly. And so you also find it actually there's in some hosts, so you have to sort of, but luckily, so as part of some of the RNA processing, the small RNA pathways that are involved in sort of sculpting or regulating some of our RNA biology, but those ones are so different. They're easy to sort of identify and sort of push aside. But what you've got here is sort of this central feature of RNA virus biology. And so that gives you good confidence that if you identify one that isn't sort of in the eukaryotic sort of family, that you're on the right path. I mean, it raises a question, and I think the authors point this out too in some related work that there are certainly probably RNA viruses out there that don't have RDRPs, and those are all of these methods would be invisible to that. And so, sort of these layers of the onion of the virosphere that we're continuing to employ, but already just using this as a signal is in real time revealing all kinds of virus diversity that we've just barely started to glimpse at and certainly don't understand yet from a virological standpoint. Okay, so what I think this paper is nicely doing, or the preprint's nicely doing, is it's using that sort a really targeted analysis of those 18 genomes to get at a very specific question. Are there other viruses besides the one of Vivax that infect Nolsey, which, and honestly, I think they would have been happy to find one in falciparum as well. They haven't yet. And maybe that's another point here is, you don't just pointing these techniques in any direction doesn't mean that you'll necessarily find something. And so you sort of get what you get. In this case, the sort of blinking light was for the Nolsey samples where they find and propose a new RNA virus. In the meantime, this group, so Meng Shi, who's one of the co-authors on the paper, just published or have in press a really interesting paper in the journal Cell. I thought about doing that one instead of this one. It's a more global look at the virosphere using these artificial intelligence tools, but I'm not sure it's open access yet. And it's also sort of a much higher 30,000 foot view. And so the thing that really captured me about this one is that it takes a little more narrow or incisive view, and also raises the important point that by understanding, as we've already pointed out, by understanding or discovering the viruses of these parasites, this might help us intervene in ways that could mitigate infectious disease, that could mitigate human suffering. Okay, so but I do want to just spend a minute on this. So this tool that they developed, they call it LeucaProt. And so Luca, we've been talking about Lika recently, the last eukaryotic common ancestor. So far, LeucaProt, this program, they've deployed it again to try to find these RDRPs, but maybe hinting at, actually, I don't know the backstory on how they named this thing. So Luca is in evolutionary circles is well known as the last universal common ancestor, and maybe they're making a case for RDRPs in sort of these very early primordial soup kind of days at the origins of life. And honestly, the evolution of the origins of RDRPs is another fascinating topic that I imagine we'll revisit at some time. Marilyn Rusnink, a great plant virologist, recently retired, has a really interesting paper where some RDRPs from some plant viruses that they were following also appear to have reverse transcriptase activity. And how RDRPs relate to another enzyme called telomerase, which probably has some origins in these sort of transposable element spaces, is really fascinating. But maybe beyond the scope of today's discussion. Okay, so LeucaProt, the way this works, which is new to some degree, and there's some other competing tools, but the way this works is, so it's using really sensitive algorithms to pull out even just sort of faint sequence identity in ways that aren't super obvious from sort of traditional approaches, and to train the software to try to learn, quote unquote, learn these patterns, and then using training sets. And so I think they feed the software 6,000 known RDRP sequences, put that into a collection of 200,000 plus non-RDRP sequences, and then as the researchers give new sequences, and millions or billions of these sequences, can this thing then sort out or propose what's an RDRP based on sort of the training set? And so it's not just the sequences here, but the advances. And we've talked a little bit about this, but so structural predictions, which is also based on another sort of set of machine learning. So things like AlphaFold, where you can take genetic sequences, RNA sequences, in this case, that encode for RDRPs potentially, or anything else, make a folding prediction and then compare the structures. And so, especially with viruses, the sequences, the letters, right, of the code, the amino acids, will diverge really quickly, but the protein, the RDRP in this case, might fold up and have a 3D structure that basically, that's the sort of the thing that propagates through time, through the generations, is that structure stays the same even though the sequence is different. And this works because we're still learning about the sort of principles of protein folding that are beyond sequences, but there's many ways to make folds that sort of match or sort of have that durability, or that those functions that depend on the shape and the geometry of the protein. And so by combining these two things, sort of structural predictions and analysis with known RDRPs, all of the sequences, to just get sort of those glimmers of maybe this came from a common ancestor. The software is getting increasingly powerful and are proposing all kinds of RDRPs that would have been missed using old software techniques. Okay, so I'll probably circle back to that at the end, but basically, so they get a hit, just not doing the sort of massive every data set, but these 18, using a more traditional diamond blast search, and that's just from the Nolzi group. They don't find this in any other groups. So it's a 3,100 base pair sequence that looks like a viral RDRP. And because they have this data set, they can do a little bit of kind of, surface level epidemiology here. So of the patients, of the 18, so the two samples where they pulled that virus sequence from, they were actually from nearby villages, and had, sort of in both time and space, there's sort of a correlation or connection there. Whereas the other 5 that don't have it were sort of more dispersed. And they also had higher parasite loads than the other patients. So this gets to your point from before, Vincent. It was, the idea is if there's also a virus involved, replicating in that population of parasites, that might influence the immune response, which could then cause inflammation, which could then cause the parasite to do better versus the virus to do worse. There's sort of it gets complicated in a hurry. So I wouldn't, you can't put this is all correlations. They haven't, they don't have the virus domesticated. There's very little virology here. It's just one sequence so far, but really illustrates how kind of building that circumstantial case, you can sort of make predictions about what's going on, and it sort of guides the research into the future. Okay, so the sequence it looks like a really distant cousin of that other Plasmodium virus, the Matryoshka RNA virus 1, in this sort of just really emerging class of these mycoviruses. And so it's not like a completely new discovery. It's still in that class, but it's it's a pretty good advance, honestly. So,
but first in this species of Plasmodium, right?
Exactly. And so it's sort of bringing a new, like a class that's barely studied, but extending its range. And here's where there's just this really cool, so almost it feels like a random connection. So mycoviruses, fungi. So we're talking about mushrooms. So it turns out there's other researchers, totally different work, not thinking about, patients in a hospital, but are collecting things like mildews and mushrooms and other fungi, and have found mycovirus sequences. And so the authors point out that and published that, shared those sequences with the world. And so the authors point out that there's that study gave them, again, that comparison point. So there's other viruses in this class, and it allows them to start to categorize this into different lineages. So even sort of more distant cousins, and these really cool sort of host range situations in species that we never kind of put together, but are together in ecosystems. So a lot of these parasites living in soils or other, sort of terrestrial habitats, are in contact with fungi. And so, there's sort of this implied connection or evolutionary history of this class of viruses that have diverged quite a bit for these different hosts, but that, sort of show a whole nother sort of genetic conversation between hosts in this class of viruses. Okay, so they search. The next thing they do is ask, how prevalent is this thing? And so there's plenty of data sets out there. I think they, there's a library of 1,500 or mycoviruses from this group. And also, so 1,500 data sets of Plasmodium knowlesi, and they ask, how many more of those do you see this virus? And the answer was none. So that's, they kind of got lucky here that they found it in their 5 samples as that relates to the next 1,500 they looked at. And this is another, I think, important theme of virus discovery, is sort of the serendipity of finding something, like kind of being in the right place at the right time, because these virus infections sort of can move in ways either like geographically distinct or temporally distinct. And so just because you find one here doesn't mean that you'll continue to, or that it will be like sort of infecting everything. And that doesn't mean it's not prevalent. It just means this is really dynamic. And so capturing these snapshots can be a real challenge. And so another, I think, sort of fun detail of this study. Then they can do this wider, cast a wider net and look for other viruses of fungi, so from this other group, and sort of put it into a little more context of how these things are all related. And I think they proposed 3 major lineages: alpha, beta, gamma. And this one, I believe, is an alpha. The gamma are a lot of viruses that were, or sequences that were found in fungi. And then it gets even more wild here, Vincent. So there's another player here. So this is, biology is, there's always more layers, or there's more complexity, or interfaces or interactions that are sort of right under our noses. So
yeah,
remember,
hence Matryoshka, right?
There you go. That's right. And so in this case, the other very obvious player are the mosquitoes, the arthropods, right? And so Plasmodium, the Cosmolaria, have this complicated life cycle where they hang out in mosquitoes. There's a blood meal. They get into our bloodstream, get into our red blood cells, take over shop, go through a whole different part of their, sort of sexual and asexual reproductive cycle. I won't be able to do it justice here on our podcast, but then get back out into mosquitoes to transmit. And so, so then the question becomes, and they already have some pretty interesting sort of signals that these mycoviruses are sort of hitching a ride. Are they, both potentially on the arthropods as well as the parasites? And the reason they get to that is that they find mycoviruses in other arthropods. So things like, insects and sort of crustaceans and critters that you find with exoskeletons, et cetera. So now they, so they search, I think they found 15 more, but just by taking the sequence that they discovered from knowlesi, looking again in bigger databases, and they find 15 more viruses in the class that are associated with arthropods. Now here comes like a cautionary tale about virus discovery. So distinguishing, so the data sets, the metagenomic data sets, just a jumble of sequences from everything that you've collected: a water sample, blood sample, whatever it is, soil sample. And so you might find viruses and you're thinking about one of the constituents, the human, if it's a blood sample, or, the Plasmodium, or the mosquito, if it's an arthropod sample. But these a lot of times these viruses can be the fleas of fleas, right? So it's infecting the thing that's hanging out on the creature. And so the obvious case for Plasmodium is that the, or mycovirus. And actually, sorry, we missed. So they named this thing. That's one of the fun things about discovering something new, is you get to name it. Do you have that at your fingertips, Vincent? I think it's
Yeah.
Yeah.
Hold on. I was just there. Oh, wait, it's down further. Hang on.
So the Matryoshka virus is the first one. The Russian dolls, right? The layers after layers.
Yeah.
Um,
It's
this
cell
one,
and dung RNA virus 1.
yeah, and cell and dung. They just have a quick note, like half a sentence, that means like concealed. I think in one of the languages in Malaysia, concealed in this case, it's hiding out in Plasmodium, and sort of under the surface. Kind of a fun
And
naming scheme
Nels,
there.
what to point out, they often use the word putative virus, right? Because
Yes,
this is just a sequence, a genome sequence.
yes,
And in fact, I wanted to ask you about this.
yeah, yeah, yeah.
They say it's bisegmented, right?
Right.
And they pull out another segment which has homology for another protein of these microviruses, right? So they say, aha, it could be the. But I don't know that. I
Yeah.
think you need to get the real virus and see if there's two segments in it, right?
Yes, and I couldn't agree more. Um, and so the difference between gazing at these sequences, it's actually given all of the complexity in the sample. It's already pretty impressive to link it to. So there's another segment separate, like not connected in the
Yeah,
RNA
yeah,
that they need that, in order to understand the whole virology here, you need to put those things together. That's clear. Whether they have that nailed down is another question. And
yeah.
whether you can get your hands on it, can you domesticate a virus? That's huge.
It's
So
hard. It's
yeah,
hard.
and that's hard. And the irony here, right, is we're talking about this virus discovery. There's very little virology here. This is all computational advances,
Yeah,
glimpsing and classifying, making inferences, correlating it to the epidemiology. But we're again, this is just like the seed or being at the outset of this research project. And this is basic science. Like it's so good. It's so like it brings together the creativity, the collaborative nature, the advances, the technology in ways that it's not clear today. How is this going to lead to a better treatment for malaria? We don't know, but time and time again,
yeah,
going down these paths, following our noses, just being curious gets us a little bit closer to something that someone will then be inspired to bring across the finish line, that will lead to that sort of translational advance. But without all of this, it might, like, we can kind of glide on fumes, scientific fumes, for a few years. I don't know if that's 4 years, I don't know if that's 8 years, or what it is, but we're eating our seed corn. We're not planting that
yeah.
seed for the future. And so this is the delay. It can feel almost imperceptible in that moment because, oh yeah, we still have all of these, new drugs in phase 3 clinical trials, but we're robbing future scientists, future generations from the opportunity to do something really interesting to sort of build off of this exciting work that's happening today. And that to me is sort of, that's the take
So
home.
basically, what you're saying is the Holmes group does the bioinformatics and say, hey, this could be a
Yeah.
virus with some interesting, and maybe someone else at some point will actually isolate the virus and confirm it and do other studies, right?
Exactly. And they are depending on the other people who are out there chasing
Of
around
course.
mushrooms and various things and lifting up these mysterious sequences. It all builds on, it's just this incredibly, worldwide consortium of curious people who just can't help themselves. They just want to sort of explore and understand the world around them. And the
So
impacts
Eddie
are
Holmes
incredible.
recently published a paper where they did a similar analysis and they found a flavy-like virus, right? Flavivirus is dengue, yellow fever, West
Yeah.
Nile, with a 40 kilobase RNA genome, right? So the biggest
Yeah.
flavies are like 15 kb. So this is the same size as coronaviruses, but no error correction machinery in it,
Yes.
right?
I love it.
So
Yeah.
I thought this was very exciting. They don't have virus; it's a
Yep.
putative virus. But I wrote a box
Mm,
for the new edition of the textbook
cool. Yeah.
because I think this is neat. So how do RNA genome size is limited by errors, and unless you're either segmented or have error correction machinery,
Yeah.
you don't get very big.
Yep.
And so this one may have a new way of doing error correction. It's not an exonuclease like the coronaviridae. My co-authors hated it. They said, "There's no virus. You can't put this in here." So I'm
There
like
you go.
defending it. I said, "Okay, I
Yeah.
know there's no virus, but I really think it's cool." And That's the thing. You
Yeah,
maybe if we put it here, it's going to get some future virologists excited enough to actually go out and get the virus. It was in a sea sponge, right? So not
yeah,
easy to
yeah.
do. There are no cell cultures
No,
or anything. It's hard.
that's right.
So,
Yep.
but
No, but
I
that's
agree with you. You know, the whole idea is get something out there and get people
yes.
excited, right?
I couldn't agree more. And by the way, I'm also sympathetic to the card carrying virologist view as well. That there's a lot of you can go sequences can fool you, and there's always sort of these stumbling around, these fits and starts, and so balancing those things is important as well. But Vincent, in that other paper, so this one in Cell where they're using their leukoprote, they do
Mm.
exactly another kind of deep dive into RNA viruses with large genomes. And there are distant cousins of the coronaviruses, the nidoviruses, a lot of them from like worms, planarian worms, not parasites, but other critters that are, I think the biggest one was something like 48 kilobases. And
64
you,
kb,
yeah, there you go. And so,
but they're segmented, and this
yeah,
is we don't know segmented coronaviruses. Doesn't mean they don't
right.
exist, right?
Yeah,
And
yeah, yeah.
so I was at a meeting where they presented that, and a guy, a virologist, got up and said, "I don't believe any of this until you get infectious virus." So who knows if there's some bioinformatic artifact that makes two
Right. Yeah.
segments, right?
Yeah, yeah, yeah. Yep. And fair enough. That's part of the work ahead here. Um, and doing it, you can do it. You can get fooled, but there are ways to do it carefully. And the candidates that you sort of lift up to have some confidence that these are worth pursuing. How you get there can be another sort of certainly part of the challenge. It can take years to pull off. Okay, so taking stock so far, we've got this new virus from Plasmodium knowlesi that they're proposing two segments, but it's there, so it can be really hard to put all this together. And so this is where they now fall back or add in to the conversation that new tool, that AI approach, which is now going through something like 10,000 data sets instead of 18. And so they pull out using that approach and the structural sort of comparisons of the RDRPs. Do these sort of overlay in a way that there's some of the geometry of the proteins look enough the same to classify them along those lines and to put the sequences then together in the tree? What they see is if they take that sort of massive data set and then put the viruses that they're seeing from this study, from that other study on the fungi, that they're sort of sprinkled in, kind of interspersed in those trees of the variation of these mycoviruses. And so what that suggests is that basically this is just the tip of the iceberg, that these things are all over the place, and they're only starting to get sort of little points, sampling points on the map, basically, of all of the viral diversity in this class. And so they can put it into what they're calling a supergroup. So that AI classifier, I think, proposed 180 supergroups of RNA viruses, which is just like, it's kind of an arbitrary number in one sense, but it just sort of like we're even today, as we're still seeing new things every time we sequence and being able to classify it in new ways by using these increasingly sensitive and powerful tools. It's all a hypothesis in the any tree is a hypothesis, as we've discussed on TWiEVO before, and so we're constantly updating and comparing to get a sense of how all of these genetic elements, all these viruses, are related to each other. Okay, so a few take-home points for me. One is echoing and to some degree what the authors are saying, but arthropods. So that was sort of a sub-theme that came out of the work, is that there's really a richness in the viruses that they're finding associated with mosquitoes or insects or crustaceans, other critters. My favorite example is there's a number of viruses that have a different genetic code. So what this means is that it can give you some clues about the host. So the genetic code, this is our letters, our codons, our three letters of DNA that then get converted to RNA that then are each an amino acid. For some hosts or for some species, their genetic code is different. So the codons will encode for a different amino acid. And so, this is an interesting, actually, immune defense. If you have a different genetic code, that could probably limit a virus if it or put up a roadblock where in order to make more virus, it needs to use the decoder or the ribosome to decode its codons. And if that's different, you'll sort of make junk instead of proteins. And so they find a couple what they're proposing as cilioviruses. So ciliates, which are these other single cell eukaryotes. Some of them are parasites. A lot of them just sort of swim around in ponds and are just sort of hang out. And but they're viruses of these guys that we're just glimpsing at for the first time. And the reason they're kind of making that link is that the virus, the genes in the virus genome, the RDRP, for example, it decodes the codons differently. So to make a real protein, it would have to be in a host that's a ciliate, or it would have to have the tools or encode the tools to use a ribosome that wouldn't make productive proteins.
So they suggest using diverse codes when you do the searches from now on.
Correct. Yeah. So so a lot of these searches, they just say, oh, well, it has to be the standard genetic code. So anything that doesn't sort of follow that rule is probably junk. And so we'll just throw it out. And yet I think there's like hiding right in plain sight are all of these sequences of different classes of viruses that infect hosts that have different genetic codes. The other point, I think I already kind of made it or tried to make it, was that the combination of the small scale, so the very directed, we want to look at potential viruses of Plasmodium using these 18 data sets, but then relying on that bigger context of the same kind of emerging tools to really get a better sense of the hosts and sort of the diversity of these virus classes that we're just glimpsing at and for the first time. And then finally, maybe closing on this preprint is the going from sequence to something that a lot of humans might deem as useful. Getting that foothold to actually start attempts to try to domesticate or launch this virus, to be able to grow it in Plasmodium, and then do studies to understand how does it change the symptoms of the disease, or using model systems, other animals, to see plus or minus the virus. If it's infected, does that mean that the parasite is less likely to cause disease? Is it more likely? Can we deploy these viruses in ways that might modify the disease process to favor human health?
I like this last paragraph. They say this, as with other metagenomic studies, primarily serves to generate hypotheses and raise questions about RNA virus evolution and biology that require additional experimental data to answer. These guys don't do that. They do the bioinformatics.
That's right.
Every time we talk about a bioinformatic-based paper, you have to remember there's more work that needs to be done. In this case, just to find out if there is such a virus infected and Plasmodium. And by the way, you can culture these Plasmodium, so somebody can easily see if there's a virus in them.
Exactly. And so, kind of getting that on the radar. And then that's sort of the, in the best case scenarios, it becomes highly collaborative. Different fields start to collide, use the information gained by these approaches to then advance the work, whether it's in parasite biology, virology, all of the above. And that's sort of, kind of one of the things, the exciting things about practicing science today is that there's so many tools and so many scientists coming up who are interested in crossing between disciplines and really thinking about, okay, this adds a little bit to the puzzle. This adds a little bit. How do I train to be able to take advantage of all of these things at once? And so these sort of scientific generalists are just sort of poised to really advance the field, assuming we can get the resources to them and keep those pipelines of support open and running.
Yeah,
which feels like a bigger question today than it did even 3 weeks ago.
Well, it's interesting. The authors on this paper, none of them are from the U.S. It's from Malaysia and Australia and China. Yeah. All of these countries have infectious disease research programs. None of them are going to discontinue them. They'd be stupid to do that, right? As we would be. Yeah. In particular, China, which is the enemy of the next administration, right? Mm-hmm. They're going to keep doing it because they realize that it's important. So for years, people from China have come to the U.S. to be trained to do research. They still do.
That's right.
So you're just going to throw that away. It makes zero sense. It reflects someone who has no knowledge of the system whatsoever. Zero knowledge. And you don't want that person to be the head of the system.
Yeah. Yep. No, it's well, and so one of the magical things, near magical things about science in the U.S. You're kind of getting at this already, Vincent. You know, in the last 5 decades or something, is just like you're saying, people coming from all over the world to hang out, learn, and then in a lot of cases, not just hang out but stay. And so the
Yeah.
lifeblood of scientific advances has been through immigration over near term and long term courses. And so that's sort of giving, like, if you want to sort of frame it in sort of a competitive sort of geopolitical framework, do you want to give away all this talent that comes here? Do we want to let it go, or allow technology to be advanced other places in ways that we used to do or used to be the leaders on? What does that mean for trainees if they're if you can't help yourself, if you really want to, like, so if I'm a graduate student, I'm kind of over the hill now, but if I'm someone young coming up in virus discovery, I'm thinking pretty seriously about getting a plane ticket to Sydney, Australia, and setting up shop, in and around the science that's happening in Eddie Holmes groups and others there. Like, it's just where things are really moving forward in ways that are dynamic and interesting. Still happening here, and again, as we sort of brace for what feels like an unnatural disaster, not clear what the sort of like the near term or long term impact, but the fact that it's even sort of so much in play for just dismantling or starving some of the traditional sources of support and resources is pretty jarring, to say the least.
So we have some comments that have come in since we last did it. Here's
Right.
Bobo Pokemono from Vienna.
Welcome.
Welcome. And here's the link for the write a letter to your senators, safecommunitiescoalition.org/RFK. Mark is here from Helsinki, where it's 3 degrees and snowing.
Getting dark.
Yeah.
Season is turning.
Abdulaziz, thank you for your support of science communication. We need it in these times. Microbe TV will keep going and talk real science all the time. It'll be your haven of where you can go to find that. And then Les points out that the American Samoa measles disaster of RFK Jr.'s is part of that video. You should go listen to it. It's just a terrible story and shows how misinformation can kill.
That's right. The vaccine skepticism, it sort of has an echo. We saw this evolution has been attacked in various ways, shapes, and forms. This notion of teach the controversy, it's sort of, you add more, it's seeming like semi-innocuous. I don't know if it's right or not, but I just want to explore that. I think RFK is signaling that, I don't want to block vaccines. I just want some information or more research. All under the surface of this is a perfect example of how this sort of cowardice of saying, I'm not trying to stop it, but you throw this information that leads to real consequences. And then you don't have the courage to stand up to say, actually, maybe I shouldn't have done that. You just sort of weasel around. That's the thing.
You say the measles vaccine is killing kids in Samoa.
Right.
So they stop using it and kids die. There's a big outbreak. What do you mean you don't want to stop it? Why are you saying these things if you don't want to stop? Then don't say that, because it was not the measles vaccine that killed the 2 babies. It was the diluent, which they made a mistake and put a muscle relaxer in. Killed them immediately, okay? And he kept saying the measles vaccine is killing kids. He didn't have to say that. He could have told the truth. So when he says, I don't want to stop them, I don't believe him for a second. He's a frickin' liar. He never tells anything truthful about vaccines and science, as far as I can tell.
Yeah, and then
he wrote a whole book about Tony Fauci, Nels, which is all wrong. It's bull.
Yeah. Well, and then it'll cloak it by saying, "Oh no, that's not what I meant," even though there are now
exactly
incredibly almost 100 kids dead. But it will be, "Oh no, we didn't mean that. What we meant was we should just do better studies." But the impact has already happened, and you can't put the genie back in the bottle.
Nels, eventually they realized their mistake. They vaccinated lots of kids. The epidemic stopped. And he said, it wasn't the vaccine. It was clean water and better nutrition that stopped it. What kind of BS is this?
Yeah.
I'm sorry. It doesn't stop an outbreak.
It doesn't stop.
Clean water has nothing to do with measles transmission.
No.
Anyway, we
Yeah.
have a guest from Ethiopia,
Wonderful. Welcome. Welcome.
whose name I cannot pronounce. Oh, here we go. It is Dejani Worku. Hello, Ethiopia. I don't know if we've ever had anyone from Ethiopia. Can you
No,
remember?
I don't, not in my memory banks. And it's great to have this community coming together every month from
Love
all
it.
points on the map. Yeah, really fun.
Vanity showed up earlier, or another moderator. Thanks, Vanity. She says, Nels, thanks for the follow on Blue Sky.
Yeah, there's another trend line. So science is moving. A lot of scientists are showing up on Blue Sky
Yeah.
and letting go of Twitter. It'd be interesting to see. There's, and again, sort of sort of communities that in some cases took years to form that are sort of dissolving right before our eyes. And some of the one of the, I think, big advantages of the conversations, science related conversations on Twitter, is that if you have a bigger community, the hope or the possibility is that you have, it extends the reach of the conversation. So as scientists are sort of comparing notes, who do that for a living, science curious people are also able to get into the mix, ask questions, mix things up. And it's, another one of these cases of an unnatural disaster, it feels like, where now like a lot of people, as they try to have these conversations, are interrupted by things like porn bots and other, sort of misinformation schemes that just throw a lot of sand in the works. Blue Sky is pretty, so far so good. I mean, wait a few years and maybe it will be it will kind of go through the same adaptive process that we've seen on Twitter, but it's been really fun. So I've moved over. I've spent more time now there than on Twitter, and mostly just sort of listening in on the conversations, which is my favorite thing to do on social media. But it reminds me a little bit of the energy of Twitter some years ago, where just really fun to have those informal contacts and interactions, kind of kicking around science and ideas and work that's emerging.
Joseph is here from Ontario, Canada. Welcome,
Welcome.
Joseph. Gail is from Chapel Hill, North Carolina. Heather is from Kamloops. It's up in
Welcome.
Canada.
Yeah.
Penelope's in snowy Idaho.
Yeah, we got a little snow here too. It's
Yeah.
the seasons are changing.
And Nikolay is from Bulgaria. Welcome. Nikolay.
Yeah. Glad you're along.
Les says, what would have happened with the mRNA vaccines if we did not have decades of basic research beforehand? There are many, many examples like that.
Yeah, for sure.
Absolutely.
And the technology, kind of like the sort of complexity of technology, where you solve one problem but create a new one. But if you sort of get off that train, then new problems you've created, if you don't have a solution in the pipeline, things can really get out of balance in a hurry.
Alan writes, scientists and graphic artists are flocking to Blue Sky. I'm following Nels and Brianne. So
thanks, Alan.
I haven't yet joined Blue Sky. I used to be. So I joined Twitter at the beginning, folks. I joined in 2008. I have almost 200,000 followers. I don't engage anymore. I used to a lot, but it got so toxic that I said, to hell with this. But we do post our podcast there because I think even though it's a toxic crowd, they need to know, maybe, who knows, maybe they'll watch one of them. You never know. But I don't engage there anymore. So maybe I should go to Blue Sky and try having the old conversations that I used to have before it got ridiculous.
Big recommend. It's actually a nice, at the moment at least, it's a really nice positive place where there's good things being kicked around. And it's a good time to have a foot in both camps, I think. There are reasons not to let go, to try to extend the reach of the conversation, to reach more people. But there's some reasons even healthy reasons to start letting go at Twitter.
Okay, now let's wrap it up with some picks.
Let's do that. So my pick, I saw someone, speaking of social media, I saw someone post, times are bad when liberals start posting poems on social media.
That's good.
Guilty as charged, I guess. So I have one. I haven't posted it, but I wanted to share it with our crew. It's from the poet Wendell Berry, and I won't read it, but there's a link to this. It's called "The Peace of the Wild Things," and so a really, I think, eloquent way of as we're dealing in spaces that can feel like despair or grief, to sort of move towards grief and engage, be present and live through sort of the process and ways of connecting with nature through the words of this poet, The Peace of Wild Things. And so there's actually an animation that I think is worth watching. It's very short, like maybe a minute or so. But yeah, that link to the YouTube, and there's the poem there. So yeah, that's my pick of the week, Vincent. How about you? What's your science pick of the week?
So I have something to do with Luca.
Yeah, cool. Yeah.
This is an article in Quanta magazine. Let's share it here. All life today on Earth descended from a single cell. Meet Luca. So we've been talking about Luca and Lika a lot lately, and this is a layperson's explanation.
That's great.
And they refer to a study that was. I don't think we talked about this.
Yeah, we were a little less ambitious. We were.
This is nature, ecology, and evolution: the nature of the last universal common ancestor.
Yep.
And its impact on Earth.
One rung or two later in deep evolutionary time, Lika, last eukaryotic common ancestor, versus Luca, last universal. So the very kind of primordial or sort of first cells emerging from the soup.
So this is a straightforward explanation of how the work was done and what it means. If you can't, and some good graphics here, a portrait of our ancestral cell. Meet Luca.
Go to that tree. So there we can see right below there, if you scroll down right there. Yeah. So Luca is at the base, and then we were up there on that green branch in the upper right.
Lika.
And they've got that, looks like that sort of magenta branch. There's the bacterial wing, and it sort of connects through, and it's coming out of the LACA, the archaeal tree, which
the archaeal and LBCA, which doesn't quite roll off the tongue.
That's right.
It's the last bacterial common ancestor. That's pretty cool. Yeah.
Very cool. Yep. We've definitely been kicking around some of that work when we think about deep evolutionary time and the origins of life. And that's great.
And Quanta's open access, so you can go read it. It's a good read.
Beautiful.
All right. That's TWiEVO 108. You can find the show notes at microbe.tv/TWiEVO. If you like our work, we need your support. We don't do ads. We depend entirely on listener donations to run these programs. And if you think they're worthy of continuing, go to microbe.tv/contribute. We're now in our November-December fundraising period. Mm-hmm. So if you go over to that page, in addition to the usual places where you can donate, we have some incentives like stuff we're giving away.
Cool.
How PBS does. You give so much, you get this gift. So we can give gifts too. So go check that out.
TWiEVO tote bags could be on the menu here.
Nels Eldy's at cellvolution.org. Are you L. Earlybird on Blue Sky?
I am, yeah. L. Earlybird, the origin story of that one is I had a friend who didn't know whether to pronounce my last name Eld, or Elda. And so he just went with L. Earlybird because that was easy to pronounce. And I've
L. Earlybird. That's very creative. I like that.
Used it ever since. Yep.
I'm Vincent Racaniello. You can find me at microbe.tv. I want to thank our moderators today, Les and Vanity Nutrition and Barb for coming here. This is a pretty nice crowd, not too much work for them to do. And I want to thank all of you for coming. Tonight we have Office Hours live stream at 8 p.m. Eastern. It's just me, but we're going to talk about RFK Jr. Extensively. I'm going to pull up a lot of things that he said, and we'll discuss why he's off and so forth. So you'll find that here on YouTube if you're interested. And we'll be back in about a month, right, Nels?
That's right.
And here on TWiEVO, we have music at the end, and I'm going to turn it on now, ending music. That's by Trampled by Turtles. You can find their work at trampledbyturtles.com. Been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious.